For training purposes and video clarity, this was filmed outside of a certified lab. All procedure must be completed under a fume vent. Before beginning, be sure to sanitize your area using a 10% bleach solution. Step 1. Label a spin column and two 1.5 milliliter elution tubes. One is for the lysis and one is for the elution. Step 2. Add 400 microliters of lysis buffer to the elution tube. Then add 300 microliters of patient sample stored in transport media to labeled elution tubes containing the lysis buffer. Gently invert tubes 5 to 10 times. Step 3. Let these sit at room temperature for 5 to 10 minutes. Step 4. Add 400 microliters of isopropanol to each elution tube containing the sample, then close the cap tightly. Invert 5 to 10 times and place a labeled spin column into the collection tube. Step 5. Transfer 600 microliters of sample from the appropriate elution tube into the corresponding spin column and then close the cap. Step 6. Centrifuge the column at max speed for 1 minute. Make sure that you place the centrifuge cover on before beginning, and always be sure to equally distance your sample placement inside the machine. This ensures centrifuge balance. Step 7. Discard flow-through liquid and transfer the remainder of the sample from the elution tube into the spin column. Step 8. Centrifuge the column again at max speed for 1 minute and discard flow-through. Step 9. Add 700 microliters of wash solution 1 and centrifuge the column at max speed for 1 minute. Discard your flow-through and make sure that the ethanol has been added to wash solution 1 before use. Step 10. Transfer the spin column into a new collection tube. Add 700 microliters of wash solution 2A. Then centrifuge the column at max speed for 1 minute, again making sure to discard the flow-through. Repeat this exact step with wash solution 2A one more time. Step 11. After discarding flow-through, centrifuge the empty column at max speed for 1 minute to dry. Step 12. Carefully remove the column from the collection tube after centrifugation, making sure that the column does not contact any of the flow-through liquid. Place the column in the correspondingly labeled 1.5 milliliter elution tube. Step 13. Add 60 microliters of nucleus-free water directly into the spin column membrane. Be very careful here not to touch the pipette tip to the membrane. Let sit for 1 minute upright in the tube rack before moving to the centrifuge machine. Step 14. Centrifuge column at maximum speed for 1 minute. Again, always be sure to balance the centrifuge. Step 15. Remove and discard the spin column. The purified RNA is in the elution tube and ready for immediate RT-qPCR analysis. Step 1. Dispense 15 microliters of the master mix into each PCR strip tube. Step 2. Transfer 5 microliters of the nucleus-free water into the first PCR tube, then close the lid tightly. This is your negative control. Step 3. Transfer 5 microliters of each isolated RNA sample into individual qPCR tubes. When transferring the samples, be sure to pipette 2 to 3 times to mix the sample before transferring into PCR strip. Leave the last qPCR tube with no RNA to add a positive control. Please note that the positive control DNA is highly concentrated, so care should be taken to not contaminate other samples or your work area. Step 4. Gently mix each qPCR reagent tube by inverting 5 to 10 times. Then add your qPCR tube into the centrifuge and let centrifuge for 5 to 10 seconds. Samples are now ready for analysis in the GeneCount Q16 or Q96 device.